Good afternoon, everyone. Thank you, Dr. Frick, for the introduction, and thank you, everyone, for attending. Today, I will talk about one of the chapters of my thesis, entitled Assessment of Select Compounds on In Vitro Ruminal Fermentation and Their Potential for Methane Mitigation. So I believe we're all aware of the impacts of methane emissions in our atmosphere, and that ruminant animals is one of the sources of methane emissions. And this is a natural process that occurs in the rumen of these animals, and it is also an important process because it works as a hydrogen sink. And this process is driven by this specific type of microbes that are the archaeomethanogens that are able to utilize the hydrogen to reduce CO2 and then producing CH4. And this is the most utilized pathway, that is the hydrogenic trophic pathway. But besides this importance in the rumen, this production of methane, it is also correlated with losses of energy for the host. So it is not only important to reduce methane, but to decrease the impacts on the environment, but also to increase the animal productivity. There are many strategies, as Guillermo talked about. One of them that is the selection of animals, lower methane producing animals that can decrease the methane emissions from dairy cows. One of the other strategies, it is the diet reformulation. But today I will talk about the inclusion of feed additives in the diets of these animals to address this problem. There is a long list of additives that can be used to decrease the methanogenesis and improve animal productivity. Among them, ionophores, nitro compounds, nitrate, and secondary plant compounds like essential oils and tannins and saponins. And in this previous work that I developed during my master's, I tested different types of additives and their combination. And when they are tested isolated, they could decrease the methanogenesis, but the strongest decrease that we saw was when they were in combination by decreasing the methane production in 30%. Today I will focus in nitro compounds that are these molecules represented here that are short-chain nitro compounds. And among these compounds we have the 3-noxypropanol, that is the 3-NOP, that was recently approved by the use in lactating cows. And these compounds, they can act in different ways. One, it is working as a sink of hydrogen in the rumen, and the other one, that is the case of the 3-NOP, it is by targeting the MCR reductase enzyme, that is an important enzyme for the methanogenesis pathway. In this work, presented here by Ristrov, they tested different doses of 3-NOP, a low, medium, and high dose, and regardless of the dose, they saw a decrease in 30% of the methane production in dairy cows. And with this, I want to talk about the objective of my work, that was to select chemical compounds with effective methane-lowering activity and explore potential interactions and beneficial effects on ruminal fermentation of the top candidates for methane mitigation. And the hypothesis of this work was that nitro compounds would have the highest anti-methanogenic activity, and in combination with other compounds could potentially potentiate the methane mitigation properties. So, as I mentioned in my title, this was in vitro work that followed this workflow. So all the phases that I will talk about will perform in this way, where I used rumen fluid as media combined with a buffer, and was incubated with 30 mL of rumen of this media with 300 milligrams of TMR, and added the treatment. The TMR was used as substrate, as I mentioned. All the incubations were performed in triplicates, as well the treatments, and the control was with no compounds. I tested 11 commercial compounds with a purity greater than 98%, and they could possibly work as a sink of hydrogen. And the compounds that I tested were plant secondary compounds, this one listed here, the nitro compounds, and other compounds. The experimental design followed this scheme here, where the 11 compounds were tested at a single dose of 10 millimolars. That was the screening phase, followed by a dose potency, where we selected 4 compounds, 1 non-nitro compound and 3 nitro compounds, to test them in lower doses than 10 millimolars, and a final phase that was the combination of carvacrol and 2-nitroethanol in the dose that selected from the phase before. All the variables that we measure was gas production, the gas composition concentration, VFAs concentration, and rumen pH, and the data was analyzed using the linear model in R. The models were adjusted for each experiment, and the Bonferroni test was applied for correction at a 5% of significance level. So the first result that I want to show you today, it is regarding in the first phase that I mentioned, the screening phase, and this it is the total volume gas production. I want you to pay a closer attention in the colorful blocks plots. The gray ones didn't differ from the control, and the first 4 were the nitro compounds that I included, and they decreased total gas volume by 20%, and carvacrol decreased by 90%. And when we take a look in the concentration of the gases, what we can see here is that the nitro compounds, besides that didn't decrease too much of the total gas production, they were able to decrease up to 90% of the methane concentration, as well as carvacrol. And when we take a look in the hydrogen concentration, mostly of the compounds didn't differ from the control, but the inclusion of nitroethane and 2-nitroethanol increased by almost 1,000-fold the concentration of hydrogen. And here it is the VFAs concentration, where again the colorful ones are the ones that differ from the control, and carvacrol decreased by up to 50% of the acetic acid concentration, and the same result was shown in the propionic acid concentration. And on the other hand, the nitro compounds numerically increase the propionate concentration in 16%, and thus decreasing the acetate to propionate ratio, in contrast with the carvacrol that increased this ratio. Moving to the dose-dependent effect on gas production, here what we saw, the most important result here was that when we increase the dose, we also increase the anti-methanogenic activity, and we defined that the best dose was the 2.5 because we still see a decrease in the methanogenesis, but when we look to the total gas volume and the hydrogen concentration, they didn't differ from the control, so we could, that's why we select this dose to follow up. And similar with what I mentioned before, the inclusion of carvacrol decreased the acidic and the propionic concentration when we increased the dose, but again, with a 2.5 dose, we had better results. And for the last phase of this experiment was the combination of carvacrol and nitroethanol, different from the workflow that I presented before. This was using the N-consistent that we were able to measure the gas production over time and the gas kinetics. And we also collect samples to analyze the microbial taxonomy. This is the main results, and it's basically summarizing everything that I presented here about how it impacts in the methanogenic, the methane concentration. And we tested 2 doses, their individual effects, and also in a half dose of 1.25 millimolars, and in a full dose of 2.5 millimolars. And this is basically the overall results and some of the possible mechanisms of action for the decrease in methanogenesis. So for the 2-nitroethanol alone, what we could see is that it decreases the total gas and, more important, the methane production and increase the hydrogen concentration. And this could be explained by a specific target in the methanogens archaea. Specifically this one, for example, the methanobacter, that is an important methanogen in the rumen, and utilize the hydrogen-neutrophic pathway that I mentioned before. And on the other side, it is decreasing the methanogenesis, possibly by targeting this specific group, but it is not showing negative impacts in the fermentation outputs. There is a decrease in the total VFAs and the acetic acid, but there is an increase on propionic acid, and it didn't change the in vitro dry matter digestibility. And for the carvacrol, we could see that there is a decrease in the methanogenesis, but it's probably not targeting specific the methanogens. And why I believe that this is what's happening is that the decrease that happens was not only in the methanogenesis, but it was actually an overall decrease in the fermentation, because as I mentioned, it decreased the total VFAs, it decreased the acetic acid, it decreased the propionic acid, and also decreased the in vitro dry matter digestibility. And it is probably leading to a decrease in a change in the microbial diversity. And when we combine the effects, what we could see is that the full dose carvacrol is probably overpowering the effects of nitroethanol, because here we can see that for the in vitro dry matter digestibility, carvacrol and the full dose were not different from each other, and the effects on methane concentration, the full dose the decrease was closer to the carvacrol. And on the other side, the half dose that we test of 1.25, the 2-nitroethanol effects are more accentuated, where we could still see a decrease in methanogenesis, but with a lower impact on the in vitro dry matter digestibility. And for the conclusions of this work, so far, the effects of carvacrol and 2-nitroethanol are dose-dependent and can reduce methane production by up to 90% at the highest concentration dose. Their combination enhances methane mitigation by up to 96%. However, at a higher inclusion level of 2.5 millimolars, it also reduced the overall fermentation. And in contrast, a lower dose combination of 1.25 millimolars remained effective in reducing methane, while less impact the fermentation outputs. And for future directions to have a better view of the microbial taxonomy, we are evaluating the microbial taxonomy and identifying microbial shifts that are relevant for methane mitigation. We could possibly evaluate different doses combinations between carvacrol and 2-nitroethanol, varying the inclusion levels, explore alternatives combinations of 2-nitroethanol and other phenolic compounds, and test the efficacy in vivo. So thank you very much for your attention. Thank you for the Dairy Hub for providing the funds for this work, and I would be happy to take any questions.
Okay. We have time for one quick question. Over there.
Wait.
We're recording these.
Do you have any information on the impact of production parameters like milk production for these compounds?
No, because this work was performed in vitro and not in vivo, but possibly if we follow with an in vivo study, we will evaluate how it could impact on milk production as well.
Okay, let's give Alice one more round of applause.
Thank you.